Welcome to the CJD Foundation's virtual conference. We thank our hosting partners, Centers for Disease Control and Prevention, and the National Prion Disease Pathology Surveillance Center. Thanks also to our conference sponsors, Ionis Pharmaceuticals, Nancy Seipert, Chuck Cash, and Tom Fabian. To view earlier presentations, visit our website or our YouTube channel. Tonight, you'll hear from the second panel of scientists who received a 2020 CJD Foundation grant. Grants are funded by Family Memorial Grants and donations to the CJD Foundation and Strides for CJD. Tonight's presenters will discuss their unique approaches to learning more about prion disease and helping advance the field for improved diagnosis and treatment. If you have questions, please submit them during the presentations. At the end of all four presentations, our moderator will address questions to a panel of the four speakers. And now, to introduce tonight's speakers, Dr. Brian Appleby, Director of the National Prion Disease Pathology Surveillance Center and Medical Director of the CJD Foundation.
Thank you very much for joining us. I'm very excited to present 4 of our grant recipients. They'll be giving an overview of their projects. Keep in mind with COVID, some of these projects have not started or in their very infancy stages because of a lot of the research restrictions that have been done. We thank all of you, the families and people who have fundraised and raised this money for this very important research. And as you'll see, we have a variety of different areas of research that are certainly going to make a huge impact on the field. So thank you very much for your contributions. As Debbie said before, make sure if you have any questions for the panelists, write it in the box. We'll get those questions, and then when we get to the question and answer section at the end, we will ask the panelists the questions. I'm excited to present Fei Wang from the Mitchell Center for Alzheimer's Disease and Related Brain Disorders in Houston. He will be presenting a talk on generating recombinant human prions in large scale for structural studies. Dr. Wang's primary research focus has been on studying prion protein misfolding associated with prion disease, which really represents the prototypical protein misfolding associated neurodegenerative disorder that affects not only humans but animals. His lab has shown that infectious recombinant prion protein can be generated from bacterially expressed non-infection recombinant normal prion protein in a novel in vitro protein misfolding system containing auxiliary cofactors. The recent studies have demonstrated that recombinant pathologic prion protein shares structural features with the native pathologic prion protein, suggesting that recombinant pathologic prion protein is a good resource for structural studies of infectious prions. Lately, he has been working on developing the large-scale production system for generating high-quality recombinant pathologic prion protein in large quantities. So it's my pleasure to introduce Dr. Wang.
First of all, I would like to thank the CJD Foundation for not only funding my research project, generating recombinant human prions in large scale for structural studies, but also for organizing this virtual conference so we can share our research progress with the CJD families and our colleagues. I would like to start my presentation with some brief background introduction of the prion. The term prion was coined by Dr. Stan Prusner in early 1980s to describe the infectious agent that causes prion disease. By definition, prion is a small proteinaceous infectious particle, which is resistant to inactivation by most procedures that modify nucleic acids. So basically, prion is a infectious protein. So how could a protein be infectious and cause the disease? Let's take a look at the prion protein, or PRP. It has 2 isoforms. The normal one, we call it a PRPC, and the disease-associated one, we call it a PRPSC. So those 2 isoforms have the same composition, but only differ in their shapes or structures. The high resolution 3-dimensional structure of the PRPSC has been obtained for more than 2 decades. On the other hand, the high-resolution structure of PRP-SC is still missing, which I believe you've heard a lot from Dr. Holger-Willie's presentation earlier. But fortunately for us, there's an easy way. We can differentiate PRP-SC from PRP-SC by simply applying the biochemical protease digestion assay. Here on the right side, as you can see in the control normal brain, when the brain homogenate was incubated with the protease, the PRP-C is chewed away or digested away. And in the CJD brain homogenate, the partially resistant PRP-SC is preserved after the digestion. So we use a lot of this simple yet powerful technique to differentiate, to detect and or screen for the existence of PRP-SC. So again, back to the question, how could this happen? According to the Prion hypothesis, the PRP-SC can interact with PRP-C and convert PRP-C to more PRP-SC. So we've learned a lot about this process, but the detailed molecular level mechanism is largely unknown. Mostly likely it is because we are missing this high-resolution structural information of the PRP-SC. So the object of this project is to provide a large amount of PRP-SC in its high quality, which means high purity for the structure study. So as we all know, generally speaking, for any structural study, we need the protein in its high quality, and we need a certain amount of that protein for the structural study. And here are a few ways I listed to collect or amplify the PRP-SC. The first one is a traditional in vivo bioassay, which we inject an animal with different prions, and at the end of the incubation or experiment, we can get a highly infectious prion, but it is in the brain. We need to purify it, and the experiments typically last for months, if it's not years. So it's going to be time consuming and cost a lot. The second and the third ways are the in vitro based way. And in the second way, we still use the brain homogenate, but we don't need to house the animal. We just take the animal brain, homogenate that brain's tissue, and use that as a substrate to amplify. The resulting PRP-SC is highly infectious. We can get multiple strains, but again, we need to purify it. But compared to the first method, it's time and cost efficient. The third one, instead of using the brain homogenate, we actually purify the recombinant PRP-C from bacteria to start with, and the resulting PRP-SC would be relatively pure. We don't need to purify, or even if we do need, it's a simple step. And of course, we can tell it's time and cost efficient, but the only uncertainty is the infectivity and the stream property. We need to verify those. So here, I'm gonna tell you using this method how I amplify the recombinant prion by PMCA, or protein misfolding cyclic amplification, which is the technique that Dr. Soto invented about 2 decades ago. So the idea is the PRP-SC has the seeding ability, which when you mix the PRP-SC and the PRP-C together, PRP-SC would induce the conversion to generate more PRP-SC, and we simply apply the mechanical force to break down those newly formed PRP-SC to generate smaller aggregates. Then if we provide enough PRP-C substrate with the cycles of incubation and assonication, we would be able to see the production of endless PRP-SC. That's the principle. Here's how we do it in our lab. Here's the assonicator. Here's the PCR tube we typically use. In the PCR tube, we add like 90 microliter typically of the substrate. Then we add a tiny amount of the seed into the tube. And after cycles of incubation and sonication, those PRPC in the tube will become PRPSC. And then we can take a little amount from this tube and transfer to another tube to seed the reaction again and again. So like I said, as long as we provide the PRPC substrate, we would be able to indefinitely continue this amplification. Here's the condition I use to generate or propagate the recombinant prion using the mouse recombinant PRP. The results are here. As you can see, we were able to generate those protease-resistant recombinant prion. Of course, I need to prove you that those are indeed prions. So the first thing we did was to try to look under the transmission electron microscope to compare the morphology between our newly amplified recombinant prion and the native RML prions that purified from diseased mouse brains. As you can see, from my eyes, I would say they look very similar, but we need to make sure they are indeed behave similar, biologically speaking. So we inject those recombinant prion into wild type animal, 8 of them. All of them developed a prion disease within 140 days, and they died around 160 to 170 days, and the average survival time is 163 days. And here's the survival curve. All the control animals are still healthy, 400 days, more than 400 days after inoculation. The next is the biochemical characterization. This image or the experiment tell us that the mobility of the PRPSC after protease digestion is exactly the same as the native RML prion, and the glycosylation pattern is the same as well. Those two aspects strongly suggest to us the recombinant prion we generated or propagated in vitro by PMCA is indeed a RML strain. And here's another supporting evidence is the serial protease digestion profile. Those two curves between the native RML and the recombinant RML are very close. Of course, there's another very important comparison study, which is the lesion profile study. It is ongoing. We hope to have it by the end of next month, but we're very confident that the recombinant prion is indeed a RML strain. So here I'm showing you a cryo-EM image taken by one of my colleagues in our university using our newly established cryo-EM facility. We're very excited to put to generate more recombinant RML prion and to put on this microscope, try to obtain the high resolution structure information of the RML prions. So back to our object of this project, it is to generate recombinant human prions, which I used the similar setup using the recombinant human PRP as a substrate and various of human prion, native prions as the seed, such as the variant CJD. I tested it first. I've tried multiple conditions. Unfortunately, so far, all of them came back negative, which means I didn't able to detect any protease-resistant recombinant PRP, which is very disappointing, but that's what we sort of expected because it is hard to work with human prion. But there are a couple of things we definitely can improve. One is, of course, we can tweak the conditions more. We try more different conditions. The other one is we only use the variant CJD in our trial experiments. because simply we have large amount of supply of the variant CJD brain homogenate. We can afford to test the different conditions. As you can see, each time in those experiments, we use at least 10 microliters. So we have tons of the variant CJD. So we wanted to sort of like test the conditions which can support the amplification of human recombinant prion. That being said, we also know that sporadic CJD, when we amplify it using the PMCA, not the recombinant, but the brain homogenate from transgenic mouse, the conditions are different for variant and sporadic CJDs. So we're still optimistic. We can try different conditions, different seeds, and we're still optimistic that we can make it work. That being said, we cannot just put all the eggs into the same basket. So right now, this is still ongoing, but not working. We're looking at other options. The first one, we can do it, but it's going to take longer time and a lot of money. So we're going to rely on the second one, the one going to use the brain homogenate from the transgenic mouse as the substrate. So here is the traditional PMCA we tested, and we optimized the condition to amplify the human prions. Those are the sporadic CJD MM1 type, MM2 type, and a VV2 type. They're on the different substrate, the M substrate at 129 or the valine 129 substrate. One thing I want to point out, those experiments were done in the PCR tube, which has the capacity of 200 microliter. Usually our experiment was carried out in 100 microliter. It's a tiny amount. So to better facilitate the structure study, we need a larger amount. So we want to bump up the scale. So the idea of larger scale PMC is to use larger container. Here, I'm showing you a cup which has the capacity of 20 ml. And with this 3D printed cup holder, we can put 7 cups at the same time. And the maximum volume would be 20 ml. So, but I started this large scale format with RML, Prion, and a wild-type mouse brain homogenate as a substrate, simply because they're relatively easy to get, and we can afford the failure. But fortunately, the first experiment, the first cup worked very well, as it is very similar to what we seen with the PCR tube PMCA format. So to make sure this is not a fluke, I tried tested other cups, like 3 more cups at the same time, and they all worked very well. So we're confident this larger scale PMCA format is a robust system. So then we applied this to the human prions. The good news is they all worked, at least for the sporadic CJD VV2 subtype. Here is the inoculum or the seed, and within 2 days, it has reached its amplification plateau. And the same is the variant CJD on the methionine 129 substrate. Within a week, less than a week, day 6, it has reached its plateau. So here, typically in this cup, we use 5 ml of the brain homogenate. 5 ml brain homogenate is equal to 1 mouse brain. So if you consider this is 1 mouse instead of in the cage, now in the cup, within a week, we can collect as much of the PRPSC as we would be able to collect in the mouse, living mouse, but we need, in the cage, we need to wait for months. So obviously, this format is much more time and cost efficient. So the next step would be try other subtypes. We're very confident they all would work. also bump up even this from this one to an even larger cup. We have tested a larger cup with a capacity of a ½ liter on the recombinant prion, and it worked. So we're very confident with this larger scale PMCA format, we would be able to provide enough human prions. And the only thing we need to do is to purify it out of those reactions, which we know there's now a relatively easy way. It was published by Dr. Wadley's group a few years back. So to summarize the study, I hope my data shown here would convince you that recombinant prion can be generated using the recombinant PRP as a substrate and use the native prion as the seed, and the amplified recombinant prion actually indeed preserves the inoculum original prion string properties. Also, we are confident that the large-scale PMCA format is ready to produce massive amount of PRPC, either in the recombinant form or the endogenous form, which we believe would facilitate the biochemical, biophysical, and structural studies of all the infectious prions. To conclude my presentation, I would like to thank again the CJD Foundation for funding my research project. Of course, all the families who have contributed to the grant, which made this project possible. In the end, I'd also like to thank my colleagues for having supported and helped and collaborated with me to move this project forward. And thank you very much for your attention.
It's my pleasure to introduce Dr. Rodrigo Morales of the Department of Neurology in the University of Texas Health Science Center at Houston. Dr. Morales obtained his Bachelor of Science degree in biochemistry from the University of Chile and also a PhD in the sciences from the same institution. His PhD work, performed completely at the University of Texas at Galveston, focused on the prion strain and species barrier phenomena and prion diseases and the prion-like transmission of Alzheimer's disease. His postdoctoral studies were performed at the University of Texas Health Science Center at Houston with Dr. Claudio Soto, where he was appointed assistant professor in 2012 and now associate professor since 2018. His current research interests include the strain variation and interspecies transmissions in prion disease, the dynamics of environmental chronic wasting disease contamination, the prion-like features of A-beta in Alzheimer's disease, and the interaction of different amyloidogenic proteins as a disease mechanism. Dr. Morales has authored more than 40 articles in the field of protein misfolding, and I would like to add that most of these are pretty novel concepts or cutting edge. So he does a lot of exciting work. And today he's going to be presenting on exploring the zoonotic potential of porcine-derived materials from animals exposed to infectious prions. In his project, he plans to analyze the presence, quantity, and zoonotic potential of prions in porcine meat, pork meat basically, collected from animals exposed to scrapie, chronic wasting disease, and bovine spongiform encephalopathy. Dr. Morales, thank you very much for joining us today.
Well, first of all, I would like to thank the Creutzfeldt-Jakob Disease Foundation for funding this research. The title is Exploring the Zoonotic Potential of Porcine-Derived Materials from Animals Exposed to Infectious Prions. And this specific research is part of a larger program we are running in my laboratory, trying to assess the potential role of different animal prion diseases. So this is the disclosure for this presentation. And as you may know, prion diseases affect humans and animals. In humans, we can consider prion diseases as rare diseases. The incidence is low, affecting, in average, 1 person per million per year. And the most common disease in humans is Creutzfeldt-Jakob disease in its sporadic form. But in animals, prion diseases can reach epidemic proportions. For example, the bovine spongiform encephalopathy or mad cow disease was a big problem, especially in Europe at the end of the last century. scrapie affecting sheep and goats also can have a very high prevalence. And an important problem, at least in the United States, is chronic wasting disease that affect captive and wild cervids from different species. So one of the main concerns regarding prion diseases involve their zoonotic potentials. For example, it's well established that BSE or mad cow disease can generate what we call variant Creutzfeldt-Jakob disease in humans. And it is hypothesized that BSE was originally generated by sheep scrapie, although this is not completely proven. But on the other hand, sheep scrapie is also considered to be not so much of a threat for human disease. Some evidence suggests that some specific strains of scrapie could affect humans, but this is still under investigation. So there are some, well, not some, but several paradigms involving the zoonotic potential in the interspecies transmission of prions. Like, for example, what happened, for example, if the BSE material comes back to sheep, this has been experimentally done, and it's known that the strain, the agent that is generated, is different from scrapie. And the properties of this material has unprecedented features, and this is some of the things that we are investigating in the laboratory. Well, also, as you may know, the central event in prion diseases involves the misfolding of a normally folded protein that all of us produce in our bodies that we call PRPC, prion protein cellular, in its cellular form, that under certain circumstances change its conformation, it misfold, in what we call PRPSC from prion protein scrapie. So the process of protein misfolding involving the prion protein follows this kinetic. So the limiting step for the misfolding is the generation and the stabilization of a small aggregates that have this abnormal conformation. Once these particles are stabilized, they will start to recruit normally folded protein into the growing aggregates, and these particles actually will get together and they will deposit in the brain of affected individuals, generating eventually prion diseases. So we know that the limiting step, again, for the generation of misfolded prions, the accumulation of misfolded prions in tissues, it can be substantially decreased if we add preformed aggregates or seeds into the system. And actually, this explains very well the process of infectivity that we observe in prion diseases. So in order to investigate this central event in prion diseases, the conversion of the prion protein and subsequent aggregation, we have several tools. One of the best is the use of animal models, specifically transgenic animal models. Basically, in here, what is done is to remove the endogenous prion protein from mice and to add prion proteins from different animal species. This is great because it allows us to model interspecies transmission in these animal species. And by doing this, we can analyze different pathological features of the disease. For example, the position in the brain of the prion protein, tissue damage, etc. Unfortunately, these experiments are very long. Sometimes they could last for years, meaning that we will not have a rapid turnover of data, and the experiments will be expensive. So that will limit the amount of research that we could do. So something that also could be used are cell cultures. In here, we can also express prion protein from different animal species. And in general, cell cultures are easy to maintain, they are fast to propagate, they are easy to standardize, but for the specific, specifically for prion research, sometimes they are difficult to infect. And unfortunately, they don't propagate all prion strains, and many of these prion strains are relevant, and we cannot modulate these processes in cell culture. So from a couple of decades, we count with other techniques that are cell-free techniques that allow us to investigate the conformational changes involving the prion protein in an accelerated and very efficient manner. So one of these techniques is the protein misfolding cyclic amplification, or PMCA. And in here, basically, what we do is to combine in a test tube a large quantity of normally folded protein with little amounts of the infectious particle. So when these proteins get in contact and we let them to incubate, basically happen, as I explained before, a recruitment of normally folded proteins into the growing aggregate. So these aggregates grow by the ends, and at some point, the conversion will become inefficient. So what we do, we break these aggregates and we generate new reactive ends. And we do this by sonicating these fibers. When these fibers are sonicated, we generate new free ends that will allow the additional recruitment of normally folded proteins into the growing aggregates. And by repeating this process of incubation and sonication, we can reach an exponential, we can obtain an exponential generation of infectious particles. So as you can imagine, this is great for diagnosis, but also is good to analyze several mechanisms involving prion diseases, as for example, interspecies transmission. And we can do it in an accelerated way. So in this figure generated by the group of Dr. Claudio Soto and Dr. Sandra Pritzko, what it was done is try to establish how useful PMCA is for study zoonotic potentials. Like again, in here, what they did, it was to take sheep scrapie and BSE from cow, and when you test the replication efficiency of these prions into an homologous system, so for example, using sheep scrapie in sheep PSE substrate, what it's seen is a good amplification of the infectious agent, a good detection of the infectious agent. Here, each signal is represents a positive detection. And the numbers in here represent the dilutions of the brain. So basically, we can detect brain material that is diluted 100 million times. So the amplification is great. For cow prions in an homologous substrate, using cow substrate, we can see that the amplification is even better. Now, if we try to analyze the zoonotic potentials in vitro of these prions, we can see that actually is reproduced what it has been widely accepted over the years. So for example, in a first PMCA round, we can see that sheep prions cannot induce the misfolding of the human prion protein. However, if we test cow prions into the human substrate, we can see that there is an induction of protein misfolding, although this induction is not as great as when cow prions are submitted to the homologous substrate. So this makes sense. And this result tells us that PMCA reproduced expected interspecies transmission of animal diseases in this humanized model. So we have experience working with interspecies modulation of prion diseases using PMCA. This paper that we published long time ago using hamster and mice is a proof of this, and relevant for the investigation of zoonotic potentials. The group of Claudio Soto in 2011 published this paper where he tested chronic wasting disease and the potential that it has to induce the misfolding of human prion proteins. And in here, he observed that it was possible, although CWD presented a strong barrier for inducing the conversion of human prion proteins. And in addition, it was also observed that maybe only some strains of the agent are able to do so. So we follow and expanded that research, and actually using a grant that was funded by the Creutzfeldt-Jakob Disease Foundation in 2018, we generated 4 different chronic wasting disease strains, and we observed that these 4 strains had different potentials to induce the misfolding of the human prion protein. Okay, so we wanted to take this research further, and we wanted to analyze what happened with pigs. You know, pigs are an important component of human diet. So in that sense, we know also that pigs can be infected experimentally by different types of prions, prions from cow, prions from sheep, and prions from deer. Now, it is important to highlight that no natural cases of pig prion disease have ever been reported. However, we also need to acknowledge that there are no surveillance programs trying to see the presence of prion disease in pigs. And of course, at present, there are no cases reported of prion transmission from pigs to humans. Anyways, we wanted to see, considering that pigs are susceptible of prion transmission from these different animal species, we wanted to see what are the properties of the prions generated in these animals, and also try to analyze the zoonotic potentials that they could have, and whether in the future they could represent a threat for human health. And what we observe in here is that the signature of the prion strains that is generated in the pig is different compared to the one of the parental strain. For example, in here you can see the bovine prion signature. It looks like this. This band that we call diglycosylated is the most prominent, and in the pigs, the monoglycosylated is the most prominent. Something similar happened when we have chip scrapie infecting pigs. We see also that the signature profile changes after passage in this new animal species, and something also similar happened when we use deal prions to infect pigs. In addition, the study of interaction between the different animal prion diseases and pigs is relevant in the context of chronic wasting disease, because as you may know, chronic wasting disease is the only disease that occurs in the wild. It's more uncontrolled. You know, we don't have it's more difficult to contain it. And for example, in nature, animals will be dying from chronic wasting disease. You know, they will be in the forest, you know, and some animals, like for example, wild boars, will come to their carcasses and they will feed from them. And when that happens, these animals already get exposed to infectious prions, and eventually they may get infected. And if that happens, and if these animals are, for example, hunted and consumed by humans, it could be an extra problem to consider. And also, during the last year, it has been observed that the population of wild boars in the United States is increasing substantially, and in many areas we can see a high density of wild boars and also high incidence of chronic wasting disease. So all this information lead us to the hypothesis of this project, and this is that BSE, scrapie, and CWD exposed domestic pigs accumulate different quantities and strains of prions in peripheral tissues. We are specifically focusing muscles, each one of them with particular zoonotic potentials. We plan to interrogate this hypothesis in two specific aims. The first one is to identify and characterize PRPSC in brains and muscles of prion-exposed domestic pigs. And the second aim is to analyze the zoonotic potential of porcine adapter BSE, scrapie, and CWD prions. So the experimental design for this project is actually very simple. What we plan to do is to take cow prions, scrapie prion, and CWD prions, and when these prions were used to infect pigs, we plan to take the brain, the muscles of these animals, and to test them in pig PMCA and also in human PMCA. In the first one, we plan to assess both tissues and see what is the potential of the prions generated after infection, the prions in pigs after being infected, exposed to BSE, scrapie, or CWD, and see how is the potential of these newly generated prions to propagate within the same animal species. And for the human PMCA, the idea is to test the zoonotic potential, specifically focused in muscles, that is the material that will enter the human diet. And well, as I mentioned, specifically what we want to measure are the propagation rates of the prions within the same animal species, to analyze the strain properties and see how diverse the strains that are generated in these animal species are. And again, we will focus in the zoonotic potentials, specifically in brain and in muscles. So one of the main difficulties at the beginning was to get a good PMCA system to amplify prions in pigs. And we were able to do it. This was the first part of this project. Basically in here, what you can see is that we can get a good amplification for pig prions using pig substrate, and the technique is very specific because our negative controls didn't show any signal. So in order to test how the system is working in relevant samples, what we did it was to take several samples from pigs infected with scrapie and sacrificed at different times. Specifically, we analyzed brain, retropharyngeal lymph nodes, and muscles of infected pigs. And as you can see in a first PMCA round, we didn't get any signal regardless of the good amplification that we had. In a second PMCA round, we were able to maintain our efficiency and specificity, and we were able to detect prions in some of the tissues that we were analyzing. So the technique is working well. It looks promising. We are ready now to jump to the next step of this research. Unfortunately, our lab was affected by the pandemic. We had to stop some of our work, and we were not able to receive some of the materials that we needed to do this research. However, we already received some of it. We are on the way to receive the additional ones. And our future directions are to establish the replication efficiency of BSC, scrapie, and CWD-derived pig prions in homologous substrate, again, focusing on brain and 3 muscle types, to compare the biochemical properties of PMCA-derived prions with original pure PSC, specifically focusing on electrophoretic mobilities, glycosylation profiles, and resistance to proteases, and finally to evaluate by PMCA the zoonotic potential of brain and muscles of BSC, scrapie, and CWD-derived pig prions. And we will do it in 2 ways. One, by normalizing by pure PSC quantities, but also by normalizing by mass of tissue. So to finalize, I would like to thank the people in the lab doing this work, to our collaborators for this project, but especially to the Creutzfeldt-Jakob Disease Foundation for the generous support, specifically to the CJD Foundation grant, to the International CJD Awareness Day Research Grant, to the Jeffrey A. Smith Memorial Grant, and to the Strides for CJD grant. Thank you.
It's my pleasure to introduce both Dr. Brooke Sadler and Dr. Gabe Haller from Washington University School of Medicine. Dr. Brooke Sadler received her PhD from Arizona State University in evolutionary genetics in 2014. She completed her postdoctoral training in 2019 in the Department of Neurology at Washington University School of Medicine. During this time, she worked in the human genetics laboratory of Dr. Christina Gurnett, where she was a bioinformatist. She recently accepted a faculty position in the Department of Pediatrics, though she still collaborates often with her colleagues in neurology. After her father sadly passed of sporadic Creutzfeldt-Jakob disease, she decided to dedicate some of her time towards researching the possible underlying genetic risk factors associated with sporadic CJD. Dr. Gabe Haller received his PhD in human statistical genetics from Washington University in St. Louis in 2013. His lab focuses on determining the genetic underpinnings of multiple neurologic disorders, including sporadic Creutzfeldt-Jakob disease, Chiari malformation, syringomyelia, hydrocephalus, and subarachnoid hemorrhage. Together, they're presenting on the contribution of rare variant and copy number variation to risk of sporadic CJD and pleiotropy with other neurodegenerative disorders. This is somewhat similar to what you heard with Dr. Simon Mead's talk and some of his genetic work with sporadic CJD. As you know, not much is known about the risk factors for sporadic CJD, so they are sequencing the exomes, which are the codon portion of the genome, of approximately 250 sporadic Creutzfeldt-Jakob disease patients. They plan to utilize specialized analysis programs and techniques to perform case control analyses, as well as look for associations with rare variants and copy number variants and disease status. They will also compare these results with those of other neurodegenerative diseases to look for potential overlap with the neurodegeneration process in general. For this last step, they will make use of the thousands of Alzheimer's disease, frontotemporal dementia, and Parkinson's disease samples currently available to them at Washington University. They hope to use this as pilot data to get additional funding to sequence approximately 4,000 more samples available to them through their collaboration with the National Prion Disease Pathology Surveillance Center. So Dr. Sadler and Dr. Haller, thank you very much for joining us.
I am Brooke Sadler, and this is my husband, Gabe Haller, and we are sharing our work on the contribution of rare variants and copy number variation on risk of sporadic CJD and pleiotropy with other neurodegenerative diseases. Before I begin, I'll just define a couple terms in case people are unfamiliar. So pleiotropy, from the title, just means when one SNP affects multiple disease phenotypes. And then heterozygosity is when you have one of each allele at a given position. So for instance, in the example of the M129V codon, if you have an M and a V, you are heterozygous at that position. Whereas homozygosity is when you have two of the same allele. So if you're MM or VV, you would be homozygous. So as some of you might know, sporadic CJD makes up the majority of all CJD cases at about 85%. However, unlike inherited CJD, there are a few known genetic risk factors, the most famous of which is the M129V codon in the PRNP gene. And it's been shown that heterozygosity at this locus is protective against sporadic CJD, while homozygosity for either allele confers increased risk. The genotype is also known to influence the clinical phenotype, for example, with the subtypes of sporadic CJD. And it's also been found that heterozygosity here is protective against variant CJD, so acquired CJD. Another important codon that is the E129K codon in PRNP as well, where heterozygosity has been shown to be protective against sporadic CJD in Asian populations. So recently, there was a genome-wide association study that discovered the first non-PRNP association with sporadic CJD. This was very exciting research done by Simon Mead and company. And they do find, as you see here, that there was an association with PRNP. However, they also find associations with two new genes, GAL3ST1 and STX6. I should have said, this is a Manhattan plot where each dot is a SNP, and then these are all the chromosomes. The line represents the cutoff for genome-wide significance. So for our project, our objectives were, number one, to do case control analysis of rare variants and copy number variants associated with sporadic CJD using samples that we got from Dr. Brian Appleby at the National Prion Disease Surveillance Center and 1,000 elderly cognitively normal controls that we have here at the university. And then aim two is to perform combined analyses of Alzheimer's disease, frontotemporal dementia, and Parkinson's disease with sporadic CJD to identify potential pleiotropic associated loci. As we know, there is some overlap in multiple forms of neurodegeneration. COVID did sort of delay getting our samples, but we did manage to whole exome sequence 71 of our samples. And so those were from Brian Appleby. And we just got the data back recently. And we also have recently received 325 more samples that are also from the Prion Surveillance Center. But these were sent back to us from Simon Mead, Dr. Simon Mead, who was using them as well. And we are getting 1,000 cognitively normal control exomes very soon from our friend and collaborator here, Dr. Carlos Cruchaga, who studies Alzheimer's disease, and that's why he has these samples just around.
So hi, I'm Gabe Haller, husband to Brooke Sadler. So just to start, I'd like to explain a few terms that I'll use in the next few slides. So one is codon. So codon is a term used for 3 bases of sequence that codes for a specific amino acid within a protein. The term exome refers to the portion of the DNA within an individual that encodes the proteins. So it encodes the exons of those proteins, so the exome. The word exon, which I just used, is the portion of a gene that encodes the protein. So genes have introns and exons. The introns are spliced out of the RNA, and then the exons are spliced together and used to encode the protein. And CNV stands for copy number variant. And so that refers to a genetic variant that is a duplication, deletion, or inversion of a segment of DNA. Rather than a single nucleotide, it mutates a large portion of the genome. And so I'll detail a bit of the work that we've done on the 71 exomes that we've sequenced so far from sporadic CJD patients. So far, the first step, what we've done is perform exome sequencing on these patients, DNA samples. So in short, exome sequencing is the sequencing of the protein coding sequence within a person's genome. And so that's performed by taking the whole DNA from a patient and extracting by hybridization with RNA probes that are annealed to the DNA that correspond to the exons within the protein coding genes in the genome. So we take those DNAs, we perform this capturing protocol, and then we perform next generation sequencing on the DNA. We then align the reads that we obtain from the samples to the whole genome and perform genotype calling. And now what's important to note is that the majority of genetic variation in the general population and in all people, regardless of disease status, is extremely rare. So if we look at the relationship between the proportion of genetic variation here, single nucleotide polymorphisms, abbreviated SNPs, against the allele frequency, so how common that variant is in the population, what we see is the majority of variants are in this low range that are extremely rare in the general population. And so a lot of our techniques these days using these sequencing methods is to understand the role of these more rare genetic variants in disease susceptibility. And that's again sort of displayed here. When we compare the relative effect size, so how likely that variant is to contribute to disease against the allele frequency or the frequency of that variant in the population. And the color of these boxes is sort of the power to detect an association with that variant. And we can see that at very high frequency or very high effect size, so for instance, the variants in PRMP that caused the genetic forms of CJD would fall in this range here, where extremely rare in the population, but very much causative. They have extremely high effect size. And GWAS, so genome-wide association studies, sort of fall in this category where they're high frequency, but they only confer a very mild risk increase to patients. And sort of where we find ourselves in a lot of our genetic associations is in this middle range, where we are trying to identify somewhat rare variants that contribute a little or a lot to disease susceptibility, but aren't necessarily causative. So the first way, and Brooke alluded to this when she showed Dr. Mead's GWAS that was recently published, is to do a single variant analysis. So simply comparing the frequency of a genetic variant in cases and controls, where cases in our case are sporadic CJD patients and controls are patients or individuals from the general population. And so what we can do is simply run a Fisher's exact test or a chi-square test comparing the relative frequency of a given genetic polymorphism in those two groups and derive a p-value of the probability of that occurring by chance. And so if that's very low, then the probability is significant and likely suggests that the association is real and causative. And so we performed first a single variant association analysis with all of the genetic variants we obtained from exome sequencing of the 72 sporadic CJD patients, and compared that to 2,230 in-house unrelated European American controls that we have. Unfortunately, we see no genome-wide significant association. So here on the left side, we have what's called a Manhattan plot, as Brooke alluded to earlier, is every single variant in the genome is a single dot, and its height on the y-axis is its significance. So the negative log of the p-value, so the lower the p-value, the higher the dot is on the y-axis. And the red line is genome-wide significant, so 10 to the minus 8, which is 0.05, a nominal p-value, divided by the number of variants that you've done. And so that's generally around 10 to the minus 8. And unfortunately, we see no SNPs associated at that level at this point. But we are, as Brooke said, sequencing another 325 sporadic CJD patients, and so hopefully we'll have more power in the near future. And then secondly, one of the things that we often do with exome sequencing is perform gene burden analysis. And instead of every single variant, we in fact sequence and combine rare variants across genes. So in this case, we take gene A, we take all of the rare variants and cases and collapse them and compare that number, here 8 out of 10, to controls. And we do that for each gene in the genome. And similarly, we can do that by grouping genes, where we take all of the variants from multiple genes that have related function and compare that frequency in cases to controls. And so we did that again with our 72 sporadic CJD sequenced patients and identified, although no genome-wide significant or exome-wide significant genes or gene terms, we found some interesting genes near the top that we hope may replicate in a larger data set. So these genes are PUM2, STK24, and interestingly, at the top of the gene set burden analysis, we find a gene ontology term that contains genes involved in the regulation of axon regeneration. So these are very promising preliminary results that we hope may replicate in a larger data set. So again, like the genes that we found, PUM2, for instance, was very interesting because it occurred in 6, rare variants occurred in 6 sporadic CJD cases and no controls out of 2,230 patients. And that was a very near, potentially causative, but we'll have to do a lot more work to understand that relationship. It encodes an RNA binding protein that has been shown to be a negative regulator of lifespan and mitochondrial homeostasis, and has been shown to have abnormal function and in various situations. And in general, RNA binding proteins have been shown to be the cause of multiple neuromuscular degenerative disorders. We've also found other interesting genes like STK24, which is serine 3-enane protein kinase. And we found 2 genes involved in the proteasome in which they have occurred only in sporadic CJD case and no controls, which we'll look into more in the near future. So Brooke will finish up, and thank you for your time.
So another analysis that we did is looking at copy number variation. And to do that, we use this program called Fishing CNV that allows for copy number variation in exome data, and it is made specifically for rare variation. And so our preliminary results from this analysis, I should say 72 samples, sorry. We compared our samples to 213 unrelated controls here. And we found that 3 CJD cases had significant partial duplications of MTMR2, myotubularin related protein 2. And homozygous loss of this causes Charcot-Marie Tooth disease type 4B1, which is a severe demyelinating neuropathy. And so here you can see we've just plotted the location of this duplication relative to the gene here. This is MTMR, so you can see it's it's only a small portion of this gene that's duplicated here. So there's a lot about deletions of this gene, right, that cause this demyelinating neuropathy, but little is known about the effects of duplications in this gene. So potentially could be some kind of gain of function. Again, would need to look at more people to replicate this. We did find that 2 CJD patients had significant duplications on the X chromosome, which included this gene, HSD17B10. And this gene is interesting because the brains of individuals with Alzheimer's and of animals in an Alzheimer's mouse model exhibit abnormally elevated levels of this protein. And this overexpression disturbs the homeostasis of neurosteroid metabolism, disturbs the balance between pro-inflammatory and anti-inflammatory cytokines. So this increases neuroinflammation leading to deterioration of cognitive function. So again, needs more validation, but potentially interesting. So in conclusion, we have identified a number of intriguing candidate genes with an enrichment of rare protein-altering genetic variation in sporadic CJD. We've used gene burden and gene set burden analyses on our exome sequencing cohorts, and this is a powerful approach to find genes that could confer increased risk for sporadic CJD. Rare copy number variation is another avenue to look at, and we can identify these via exome sequencing, and these also may play a role in sporadic CJD risk. Additional sporadic CJD case exomes, so these additional 325 patients that we will sequence will greatly improve our power for all of these associations that we've discussed, and hopefully find novel genetic causes of sporadic CJD. So we would like to acknowledge our collaborators, Dr. Brian Appleby and Dr. Simon Mead. Thank you for working with us to get us the samples during this trying time. And our collaborators at Washington University in St. Louis, Dr. Carlos Cruchaga, who is giving us his cognitively normal control exomes. Christina Granette, she allowed the use of some control exomes that we did use in this data. Dr. Jorge De Paula is my mentor. Thank you to him. And Dr. Linda Pike has given advice and is also my mother. And we would like to thank our funding from the CJD Foundation, specifically the Jeffrey A. Smith Memorial Grant, the Tom Stevenson Memorial Grant, the CJD Foundation Grant, and the Strides for CJD Grant. And lastly, I want to thank my father. And all of this was for him and everybody else that we've lost to this disease. So thank you.
It's my pleasure to introduce Dr. Candace Mathiason, who's Associate Professor of Pathobiology and Director of the Office of Vice President for Research, Program for Research and Scholarly Excellence, Infectious Disease and Rapid Response Network at Colorado State University. Dr. Mathiason's research focuses on the role blood and maternal infections play in disease pathogenesis and transmission dynamics. Her laboratory combines use of native and rodent in vivo hosts with highly sensitive in vitro assays to assimilate an understanding of the biologic mechanisms associated with covert transmission of infectious agents. The intent of her work is to provide basic science principles for continued efforts to mitigate infectious agents via preventative, therapeutic, and vaccine therapies. I believe her work is extremely exciting. I remember when she first presented on it in cervids, in deer and elk, to look at prions in blood. And this is a perfect example of how basic science can translate into very strong therapeutics as well as potentials for diagnostics as well. In her research, which is entitled Use of Blood-borne Prion Amplification Assays for the Detection of Human Prion Diseases and Biomarker Assessment, she'll study the use of simple, rapid, specific, and highly sensitive assays to detect blood-borne prions. Early detection of prion disease will hopefully provide a biomarker, not just for prognostication and diagnosis, but hopefully for development of therapeutic trials. So Dr. Mathiason, thank you very much for joining us.
Hi, thank you. I'm happy to provide an update of the work that we've been doing funded by the CJD Foundation in the use of our blood-borne prion amplification assay for the detection of human prions and for its use as a potential diagnostic and biomarker for prion disease or amyloid disease. We know that amyloid associated with prion diseases are found in the blood, and we know this because cervus infected with chronic wasting disease, sheep infected with scrapie, cattle infected with BSC, and humans that have the variant form of Creutzfeldt-Jakob disease, not the sporadic form, but the variant form of CJD, all carry infectivity within their blood that upon transfusion initiates and develops the disease in the next susceptible host, and that this can occur with whole blood and buffy coat. Has been to try to determine whether we can develop a diagnostic assay using an in vitro system or a system in a plate glass called real-time quaking-induced conversion. This is an assay that was originally developed by Byron Coey's laboratory. We've been using this assay in our laboratory for CWD. So this assay originally was used for tissue, brain or lymphoid tissue that was put into a glass tube in combination with salt or thioflavin T. This is a dye that actually binds into the growing amyloid fibril that we know is associated with neurodegenerative diseases, as well as a substrate, in our case, the recombinant prion protein that all goes into this buffer, into a 96-well format, and into then this fluorimeter that allows for us to amplify through shaking and then fluorescent reading to be able to determine whether in that original tissue, whether there's amyloid that again binds to the fluorescent dye and creates these fibrils that we can see upon fluorescence within the machine. And that fluorescence would look something like this, where there's enough of this amyloid that's developed over time, it crosses a threshold and allows us to be able to detect it. Whereas in a does not have the amyloid seed, you would not see any of this amplification at all. So our group, again, has been looking at the use of buffy coat. So these are particular cells within the blood component that you can see right here. It's a group of different cells that we know carry prion infectivity. Putting those cells, making modifications to this RT assay. And again, similar system, and our goal is to determine whether we can detect this amyloid versus no amyloid. And in our case, what we've been doing is we've been turning these into rates over time. It just allows us to be able to look at a lot more samples and a lot more replicates of a sample at one time. Makes it much easier on the eye so that we can differentiate between a positive sample that contains amyloid associated with prion diseases versus a negative that there is no amyloid. Now, you see a few sporadic events here, but you'll also see means for each of these samples is down here at the 0 line. In comparison to samples that are positive, this black line is the mean for each of those. So again, giving us a nice definition between a positive individual sample and a negative sample. So we've been able to use this in our chronic wasting disease system. So chronic wasting disease is a disease of cervid species here in North America, both in the United States and Canada. It's also known to be in Asia and now in Europe. And so we have experimental studies ongoing in which we're able to collect samples for CWD inoculated deer from the time that we inoculate them through terminal disease. And what that's permitted us to do is to be able to use these blood samples, pulling the buffy coat, again, the specific cell subsets into this assay. And what we've been able to find is that within 15 minutes of oral inoculation of our deer, these animals contain amyloid. This is the inoculum source that went in. This inoculum source disappears by about 72 hours post-inoculation. But the interesting piece here is that during these very early phases of disease, the asymptomatic phase of disease, we can actually start detecting nascent development of amyloid within these individuals. We've also been able to better understand what's going on with the disease process. You can see here over time that the amyloid decreases and then comes back again. And then later into disease, we can follow that over time. So our goal, of course, is wondering what's happening during this asymptomatic phase of time. Can we detect the prions? Can we detect that amyloid in these animals during that time? And of course, that would be very helpful in the development of vaccines for chronic wasting disease and other diseases that are prion related. So we've been able to see that both in our cervids infected with CWD, as well as another system using a transmissible mink encephalopathy and hamster system. So that permitted us an opportunity to ask the question as to whether or not we could detect amyloid associated with sporadic CJD or in patients that have genetic mutations for portend of prion diseases, and to look in these different types of cells. So platelets and white blood cells are all part of that buffy coat blood cell component that we know there's prion infection. Can we detect in those cells, and again, apply this then to human disease that could be used for a diagnostic or for a biomarker? And we would be able to detect that during the asymptomatic phase of disease. And again, that would be able to help us when we incorporate the use of therapeutics or interventions, and again, in vaccine development. So we had some samples in the freezer, buffy coat samples from autopsy-confirmed sporadic CJD individuals, patients. And we took samples from these patients and put them into our assay. And much to our surprise, we were able to detect amyloid in these samples, in all three of these samples, as compared to healthy age-matched controls. And that's when we came to the CJD Foundation with this preliminary data that we could detect amyloid associated with the sporadic form of CJD, and proposed looking at individuals that have sporadic and individuals that have the genetic mutation that portend prion diseases. So what is the process of this? So blood that is taken, anywhere between 10 and 20 mils of whole blood, the buffy coat cells from that blood have been separated. So what are buffy coat cells, you're asking? Well, in collaboration with Dr. Appleby, we've been able to take this blood, pull off these buffy coat. How do you get that? Through centrifugation of the blood sample itself. So upon centrifugation, you get this very nice separation of the plasma, the red blood cells. These are the cells that we're looking for and that I've been talking about. They contain cells that we know have amyloid in them that are prion positive. So in taking those cells from buffy coat, Dr. Appleby was able to send us some blinded samples, or samples that we did not know what they were, from individuals that had sporadic CJD and negative controls. And what we found in putting them in our assay was that we could detect amyloid associated with a sporadic form in comparison to our negative controls. Now, one of the things that we wanted to look at was making sure that we are detecting the target, and the target being amyloid in those samples. And so we've been doing dilutional series of these. When one does a dilutional series, one would dilute out the target, and we're seeing exactly what we think that we should be seeing in diluting that out, that we see less of that target in each of these, and that compared to our negative controls. The other piece that we're looking for is just how many cells do we need to be able to detect amyloid, and to date, using 250,000 of these buffy coat cells, we can still detect a signal. More robust, of course, looking at 1 million cells, but still able to detect at much lower numbers of cells required for detection. So where are we today? One of the things that we really wanted to do is compare sample processing. So we process samples in our laboratory, other folks process samples in their laboratory. There may be some differences in there. We just want to verify that that step is not causing us to read something that we aren't meaning to read. Additionally, we want to look at more negative control samples. Again, we just want to make sure that we are detecting the amyloid, the real target that we are looking for this diagnostic assay. We want to screen more samples, and Dr. Appleby will be sending us more samples soon that we will put into this, and we're hoping to look more at samples from those that have genetic mutations that portend prion disease. We'll also be looking at samples from other sources across the nation. And with that, I'd like to thank our collaborators at Case Western, Brian Appleby and Kyle Bickett. Kyle has been processing the samples for us. And then, of course, individuals in my laboratory, Amy Knowles and Aaron McNulty, who have been spearheading this project pretty much from the beginning. With that, I'd like to thank the CJD Foundation, in particular the James Jim Brown Memorial Grant, the Linda Morris Memorial Grant, the Kenneth Wheat Memorial Grant, the CJD Foundation Grant, and the Strides for CJD Grant for providing funding for us to do these studies. Of course, my other funding with NIH, the NIAID, and NINDS for helping us initiate this work.
Thank you very much. Welcome. We're now going to move into the question and answer session. So we will do this for about 30 minutes. You'll have plenty of time to watch the debate later on, should you choose to do so. First, I wanted to thank all the grant recipients. Really excellent science, excellent research, and it's all in the spirit of helping families to get at some of the really hard questions of this disease. I also want to point out that we now have had 3 grant recipients from the CJD Foundation grant program that have also personally been affected by the illness. So of course, that's Brooke, as you heard today, and Sonia and Eric, as you've heard before, and of course, Joel Watts. But we also have 2 research couples that are grant recipients that have also personally been affected by the disease with Gabe and Brooke. And this is a rare disease, and I think that's telling. So thank you for sharing your story, and thank you for doing the work that you do. So I'm going to start with a question for Dr. Wang. There is a member of the audience who asked where your source of variant and sporadic human prion disease came from. And I just wonder if you could clarify. You had made a statement in your talk about having tons of variant CJD brains. If you wouldn't mind, thank you. You have to unmute yourself. Yep.
Are we good? Okay.
Yep.
Yeah. Yeah. That's my bad. I'm sorry about the not very careful about the words I chose. It's like too oral. I was trying to. Because I didn't try the sporadic, we collaborated with Dr. Pritzko with the human prions, and one of her research assistant who's handling those samples told me we have a lifetime supply. So what I would say it's not, because we use tiny amount as the seed. So I exaggerated that, but no, not tons of that. But what I was trying to say, like, we can use the variant CJD to try out all the different conditions without worrying about using too much of the sporadic CJD materials, which are more valuable for us.
But I think that's a good point that you make, because we know that variant CJD is a rare disease. There have only been 232 cases worldwide. And yet, even with that little amount of cases of variant CJD, you seem to still have a fair amount of material that you need for your research.
Yeah, that part, I'm not 100% sure, because I don't handle those, samples, because in our laboratory, we have Dr. Pritko, she's handling all the samples. So simply I think we received probably big chunk of one variant CJD patient. I have no idea. I'm just speculating right now. I will communicate more with Dr. Pritzko about the amount and where the source is.
Rodrigo, that's Dr. Morales. Let's just get this out of the way.
no one likes to talk about COVID-19, but obviously that has made a huge impact on all your labs. So you had specifically mentioned it. So I was wondering if you could start us off with just telling us, how has this pandemic affected researchers and your work specifically?
Well, it has been a problem. I think that not only for us, I think that for everybody at different degrees, you know, specifically for the lab, we first were able to reduce the amount of work per people that was possible to be done. Then the university was applying more restricted measures in order to facilitate social distancing and also to try to contain the disease, especially considering that we are working in a hospital area, you know, so that was critical here. And in addition for that, as I mentioned in this work, we are collaborating with different groups. And these groups also had to close, you know, some samples where we were depending on them to get some of the samples. And what you can do, you know, and in addition to that, the uncertainties, you know, of what will happen in the future. You don't know what will happen next week. So in that sense, it's difficult to plan, you know, so what to do, what to start, because you don't know whether you will be able to finish it or not. I think that now we have a clear picture of how we can work, but I will say April, May, they were difficult months to plan. So yeah, all that delayed the work.
I think you all actually got a tremendous amount of research done, given all the limitations. So I think you're being commended on that. Anyone else have anything to share about how COVID has affected your work, or just the basic lab environment? So keep in mind that a lot of these family members might not know any of the specifics of what you had to shut down, restrictions, that kind of thing.
So we had to shut down. I mean, our university went to 25% capacity there for a while. And you know, since you sent us some of the samples, that it was quite the ordeal working with the MTAs and the IRBs during the pandemic. And then even when we did get the samples, it was not a priority because COVID samples are the priority to sequence right now. So we were kind of at the back of the line there. So you know how they say, if it weren't for the last minute, nothing would get done around here. That's kind of how it was.
Yeah, I would share with that too, Brian, that at CSU or veterinary school, and we actually got CLIA certification to do COVID testing. And so it put a real bind in what was our typical pathway from the veterinary perspective to have people start running the human samples at CSU as well.
Am I remembering correctly that you actually had a pretty strong leadership role during COVID in your university as well, right?
Yeah, yes, I have. I spent the first 2 or 3 months as the director of a collaborative group. I spent a lot of time matchmaking and trying to help people find funding for their COVID work, which is also, we have a huge infectious disease department. And so there was a lot of opportunity for folks to work together. And they did. They literally dropped their research and moved toward COVID research to try to answer this question, from anything from vaccine development to antivirals to more mechanistic understanding in animal development for models to better look at it. So, yes.
Faye, do you have anything to add? Your experiences?
Yeah, me and Rodrigo, we are actually work like in the same building or same university. Yes, for me, it's more of the substrate, because of the COVID-19, especially at the very beginning, they shut down the animal facility without we cannot get any animal, like either purchased from commercially available animals, or like our collaborators, because we use those like bring homogenate as a substrate. Of course, we restrict all the personnel, access to the building. We have to come down to like a skeleton crew, which is the essential personnel. We limited like I guess like yeah, it's about 25%. So most of people working remotely from their houses or apartments. But I was luckily I was able to continue. Like we have shifts like in the morning, afternoon. I was in the afternoon shift every day. It's a 3 to 9. But you know, like research is not. It doesn't. I mean, it's most likely 3 to 12 during the midnight. So, yeah, we have to keep working on this. So even it's difficult time, but yeah, that's all. We're all affected, but now hopefully we got a vaccine. We can have move away from this, and we are. I hope we can all get together at the next International Prion meeting. Hopefully.
Well, I know all of you were a little stressed out when Debbie started sending out the invitations for trying to get the talks together. And really, I'm impressed with how much work everyone has done, given everything that's going on. So this is a question from a family member, and this is for Gabe and for Brooke. So this family member's father passed of CJD, and on the autopsy, it showed sporadic CJD, but it also showed another illness, cerebral amyloid angiopathy, and they wanted to know if there was a link. So we can use that as a specific example, but maybe you could talk about other possible disease links that you might find with some of the research that you do.
Well, I can say that what we know about the inheritance of at least many neurodegenerative diseases is that often families have multiple different neurodegenerative diseases that run in their families, and it's unclear at this point whether or not they have a unifying underlying genetic link. That's partially why we're trying to do the work that we're doing to understand that, if possible. Maybe Brooke, you can anecdotally tell about your family.
Anecdotally, my grandfather and great-grandfather died of Alzheimer's, and my great-uncle died of parasupranuclear palsy, which is another rare neurodegenerative disorder, all on my dad's side. So yes, I agree, it seems suspicious.
So it's always possible that they could, work negatively together to increase the probability of negative outcomes. So.
Wasn't progressive supranuclear palsy another rich gene that Simon found? STX.
Oh yeah, it was. Yeah, it was on at the same haplotype. Right, you're right.
Okay.
So it's interesting to think. Also on that same note, for instance, in Alzheimer's, not to get too tangential, it's often the case that frontotemporal dementia is confused for Alzheimer's disease because they're so similar. I mean, in the case of Creutzfeldt-Jakob disease, it's so specific, and there's specific tests that we know that people have CJD. But it's possible that 50 years ago, when your uncle was diagnosed with a disorder, they actually had a different disease, and they just didn't have the molecular tools to diagnose it properly.
That's an excellent segue about clinical testing. So a lot of times, from a family member's perspective, they think of diagnostics of prion disease. They think about amplifying prions. They think about RT-QuIC, because that's what their loved one got, with the spinal fluid, and a lot of times that's how they got diagnosed. But when they hear researchers talk, they hear some RT-QuIC, but they also hear about kind of RT-QuIC's cousin, PMCA. So I was wondering if Candace, Faye, and Rodrigo, if you could talk a little bit about how they are similar, how they're different, and what's the pros and cons of each.
I think Rodrigo can go with the PMC as he's one of the original developers of this technique. I can make some like in terms of recombinant.
Yes, not original. The technique was developed by Claudio Soto a long time before I joined his group. But yeah, I have participated in several studies dealing with PMCA, and from my experience, I will say that the techniques are complementary. They have their pros and cons. From the diagnostic perspective, RT-Quick looks very nice because it's a technique that is fast, provide you results in real time, and importantly, when you want to set it in a diagnostic laboratory, this technique will generate material that is not infectious. So that's the problem from that aspect with PMCA. It's a problem and an advantage, because PMCA will generate material that is infectious. This infectivity will allow you to do studies to understand better, how the prions misfold and propagate. Well, from a practical point of view, what we have seen is that PMCA is more amenable to some samples, so to some specific strain, and RT-Quick may overlap or be better for other strains or sample types. I think that what the field need to do is to get together and try to compare results, compare the data. I think that in particular strains, the specificity of one technique versus the other is quite similar, but to see the usefulness of one or the other in terms of specific diseases, that need to be tested in a larger study involving several laboratories handling techniques. I don't know, Candace, what you think about that?
Yeah, no, I agree with that. I actually think of PMCA as sort of the father prior to RT-Quick, in that it was the first test that we were using for amplification. And then sort of the next generation of testing that came along was the RT-Quick assay. And certainly we've been doing exactly what you're suggesting, is to try to assess how do they compare? How does PMCA compare to RT-Quick and to immunohistochemistry and to other assays, to have some idea what does it all mean, and how can we speak as scientists across all these assays? Because we use both. We use PMCA, we use RT-Quick, and you're spot on. There are certain samples that it's much easier and much better to use PMCA than it is to use RT-Quick, and vice versa, for all the reasons that you say. They're both incredible assays and have brought us to very new understandings, in particular about very low quantities of material to try to better understand what's happening during those really early phases of disease, that asymptomatic time in disease, when we know individuals have amyloid within tissues and body fluids, but it was impossible for us to detect before these two assays. Unless bioassay, of course, which is a whole nother story.
Of course, clinically we use RT-Quick because just technically it's much easier. It's quicker, and you don't have a lot of the technical issues that you have with PMCA. So that's why we don't do PMCA on samples, although there may be a time in the future where, if it's a case that's atypical and doesn't react to RT-Quick, we might end up doing a PMCA. But at least right now, clinically, RT-Quick would be the test of choice. Question from another family member. This is for Brooke and for Gabe. You talked about, in your researching sporadic CJD, about common links, and you found a specific marker on the X chromosome. And they asked that since it's on the X chromosome, wouldn't it be a type of genetic CJD as sporadic CJD? So I guess we need just kind of a little bit basic genetics, how many chromosomes, what are the X and Y chromosomes, if you could elucidate out just a little bit, just for those without a genetics background.
Sure. So as you may or may not know, there are 22 autosomes, so non-sex chromosomes, and then an X and a Y if you're a male, and 2 X chromosomes if you're a female. And so mutations or variants on the X chromosome are just sort of special cases because males only have 1 X chromosome, and so often if you have a deleterious mutation on the X chromosome, it's worse in males because you're, quote, hemizygous, you only have 1 copy, and so it appears as though you have only that version of the gene. Whereas in females, you have 2 copies, and so having 1 bad copy may not be super deleterious. And so that's the only sort of caveat and why we specifically say things are on the X chromosome usually.
Right, so it might be more severe in males, but it doesn't mean it's like a genetic form, so to say. Right. Yeah.
Hope that answers.
Yeah, thank you. Everyone has different backgrounds on this call. It's it can be challenging, but also good because everyone learns from one another. One question, again, I believe is from a family member, and I think this is kind of coming out of our earlier conversation, Faye, is kind of the integrity of the tissue that we do get from patients. So they had asked, how long can a brain be viable when it comes to frozen tissue? So generally when we do a brain autopsy, we freeze half of the brain. And I can say, at least from some of the stuff that we do, the other day we used part of a kuru brain that was frozen, 70 years ago. But if you could each talk about the different research that you do, how does that age of frozen tissue impact what you're able to do with it? And then how much tissue are we talking about that you need to do the type of work that you do?
Yeah, I think because I work most of my research with the recombinant. Lately I moved to the human prion, which we work a lot with the human brain tissues. To me, just purely from science of we know prion is very stable structurally. So in terms of the age of the brain, how long we have stored at -80, -20, even though it would affect other the whole structure during all the time. For PRPSC, I would say, or the prions, they don't affect that much. That's my understanding during my study of the prions. They're really stable. So it wouldn't affect in terms of the PRPSC structure or the string property wise. But I would say, of course, because I started with animal prions, like a mouse prion string, like RML. That's the most popular one we use. It's very, for me, once I found the right condition, it's very easy to reproduce that experiment, just amplify from RML to the recombinant RML. It's really easy. But right now, for me, I just get into the human prion field. Like I said, the conditions may vary a lot to amplify those recombinant prion. So in terms of that, I think that the amount of the how much I can get to try matters. But I personally, I don't believe the age of the brain would affect 10 years versus 20, 30, 50 years old of the brain tissue in the freezer. I don't think that's because we all know for the CWD, they have go through all the environmental tests that people have published. Jason Bartz, I believe, published a freeze saw mimic the environmental, all the conditions. Prions are pretty tough. And they still have their string properties. So in terms of just time wise, I don't think that would affect. But yeah, I just need to find That's it. Yeah, that's probably you need to try multiple conditions. If you're lucky, maybe you can get it the next day. Maybe you need a couple of years to find that right condition. So, but yeah, I do not. I think Prion is pretty stable, and that's actually work better for me because I don't need to be picky on the tissues. Yeah.
What about other people's work when it comes to different tissues like blood and different material that you're looking at like DNA? I'm referring to Candace and
I'm sorry.
Brooke. Sorry, that's all right.
Well, I can tell, for example, the experience that we have with the deer samples. So we have received field samples, that has not been collected like what is done in the laboratory, with the very with more care, so the samples are carefully obtained, but they don't have the state-of-the-art equipment to collect it. So sometimes we receive samples that are thawed, or sometimes we receive some samples that are, for example, blood samples that are clotted, and at least by PMCA we can detect them anyways. Now, these techniques are very sensitive, so we don't know how much we lost. If there is something there, even if it's very little, we can detect it. So at least for the replication techniques, it works like that. But in our opinion, we have seen that, in our experience, we have seen that, I don't know, brain samples or contaminated material that have been frozen for years, the infectivity titers are reduced very little, if they are reduced. That's our experience, at least.
It looks like Candace can't unmute herself. Gabe and Brooke, are you able to unmute yourself?
Yeah, we can.
Okay.
As far as quickly, DNA is very, very stable, especially when frozen for millennia. So, I mean, people have sequenced woolly mammoths that they found in the snow. So, yeah, DNA. RNA is not so much stable, but DNA for many, many years.
It looks like you're unmuted now, Candace.
Oh, excellent. I was typing my answer. Thank you. Thank you, whoever did that. That was magic. We've been doing experimental studies with chronic wasting disease for over 20 years now. And so we've been using much as Dr. Morales is saying, we've been using those samples over that time frame. We don't use a whole lot, and so we can get by with using nanograms or microgram quantities, which is really a small amount of tissue, maybe the size of like a pencil eraser head or less in doing the studies, and they seem to be consistent over time. With blood, same thing. We've been able to freeze back buffy coat cells for time and be able to bring those out and work with those. It takes about 2 to 3 mils of whole blood to gain access to the number of cells that we need. It's better to have more, of course, but in reality, we could probably get by with less. And so it really takes a very small amount of material to get what we need for the testing we've been doing.
I think that's important for families to hear, because we often get questions about the loved one's samples, and really their loved one samples go a very, very long way for a very long time. Mm-hmm. And can contribute to multiple research studies. So I'm sure they like to hear that. Rodrigo, one question. Sorry, go ahead.
Oh, and I just wanted to ask you, how many freezers worth of samples do you have over there?
52.
Okay, thank you.
And some of them are really old, some of them are new, and they're always juggling between the old and the new. Rodrigo, you talked about a porcine model, and obviously everyone's worried about chronic wasting disease, but you picked the porcine model, but do we have to worry about other animals that may become kind of an intermediate animal that might transmit to humans later on?
That's a very good question. This is what we are trying to explore. At this moment, there are no cases reported. For example, chronic wasting disease is a concern, but no cases of chronic wasting disease transmission to humans have been described. So considering that at this moment we can say it's not happening. But on the other hand, we need to look, and in order to confirm that assumption or discard it. I will say that the case of the chronic wasting disease is particular because, as I mentioned in the presentation, this disease occurs in the wild, and there is no control of what animals could be in contact with, for example, a carcass of a deer that died from chronic wasting disease. And some studies have been made in the laboratory trying to mimic these events, like, for example, they have injected chronic wasting disease in raccoons, and other animal species, and they have seen that they can transmit the disease. Pigs are a concern, and I will say that there is worth to do some surveillance on the wild pig population, at least to, again, confirm or discard that these events are happening, so we cannot be blind to that. But at this moment, no, there are no cases reported.
So we're running out of time. I was wondering if each and every one of you wanted to just say a few final words to families, and you also have some colleagues on the call too. So we'll start with Brooke and Gabe.
I just want to say that I know what you're going through, and it sucks. But we're trying, and thank you for your support.
Thank you. Candace?
Yeah, I would echo the same thing of the support. Thank you for your support and for your confidence in us that we are working hard to try to find answers.
Hey, I see you've recovered from your power outage.
That was our dog, sorry.
Yeah, I'm in the conference room. I need to reactivate the lights. Thank you, CJD Foundation, for organizing this meeting to have we present our data and talk to the family, answer the questions, and talk to the colleagues. Knowing what we're doing is important. Of course, there's much more we can do and we need to do, but thank you very much for your support.
Yeah, well, I think that as it has been said before, thank you very much for supporting the research. We are trying to get answers, to try to diagnose this disease, and well, I really missed this year conference. I had 2 years ago, I attended, and it was really inspiring. When I came back to the lab, I came talking about how it feels to be in the conference. It's very special because it's very nice to share with the families, and it really kind of add a meaning of what we are doing, an additional meaning in addition to the science, okay, look, this is why we are working. So I hope that this everything gets better and that we can meet again soon.
Well, thank you again to the families that have contributed, not just through fundraising, but also through just helping with research through samples and whatnot, and just your strong support for the researchers in this community. And thank you, Five, also for the very good work that you do. And you all gave excellent presentations today that I think most family members were able to easily understand, and that's always greatly appreciated. So thank you for spending at least part of your career, if not the majority of your career, on prion diseases. And hopefully we do get to see each other in person soon. And with that, everyone, good night.